Hi, my name is Elisabetta Rappi from Cardiff University, and I'm here to talk about our latest study that was recently published called Restless Nights When Sick, Ectoparasite Infections Alter the Rest Activity Cycles of Diurnal Fish Hosts. I'll start with a bit of a background on circadian rhythms in general. So, what are circadian rhythms? They're intrinsic timekeeping mechanisms responsible for the cyclic repetition of many biological processes, including metabolic, behavioral, psychological, and they are found in all living organisms, typically over a 24-hour period. They're generated by self-sustaining biological clocks, which are encoded by clock genes, and they're entrained by environmental cues such as light, temperature, and so on. They have been observed in many organisms, including fish of economic importance, and they seem to govern activities from reproduction, maturation, spawning to immune responses and disease susceptibility. They've also been found in many parasites, and what we've seen is that these circadian rhythms are critical in the co-evolution of the host parasite systems, as parasites tend to synchronize their rhythms to that of the host to influence infection dynamics and transmission potential to a new host. We know that disruption of biological clocks can have a severe effect on animal health. A classic example of that is humans, where a disruption of a circadian rhythm because of jet lag or shift work or sleep deprivation in general can have a very severe effect. I'll move on to figure 1. So this is where we explain how we statistically approach circadian rhythmicity. And as you can see in figure 1, we have the 3 different variables assessed. So we have acrophase, amplitude, and measure, all in the Sitka Compare Statistics package used, but you can find more information behind the statistical analysis on the paper. I'll go on and introduce the host parasite system used in this study with a very nice picture that we have here. So the host was the Guppy Poecilia reticulata and its associated ectoparasite Gyrodactylus tumbuli. And we have a picture of an infected guppy under the microscope, and you can see the parasites present all over the skin of the host, but also the fins. So, the methods used in this study. So, in this study, we conducted 4 different experiments. 2 experiments focused on the rhythmic covariance of the host, both when infected and uninfected, both individually but also in souls. And then 2 experiments focused on the rhythmical variance of the actual parasite itself. And I briefly go through the methods used. So, we have the automated monitoring system that was used to monitor the activity of the guppies. And these are behavioral arrays, and we have the light beams that enabled us to track the activity of the guppies at different time points throughout the day, but also different light conditions. And in order to monitor the parasite activity, we were able to record the activity of the parasite present on the host at different time points throughout the day, both in light and dark conditions. So what did we find? What did the results show? For this study and for circadian rhythm studies in general, we used the Zeitgeber time system, which is a system used to measure time, basically with the first point ZT0 being when lights go on and ZT12 when lights go off, and we have different intervals between. So in this study, we refer to the different time points as the Zeitgeber time, or the ZT in abbreviation. So for the first figure, we explore the activity of fish between light and dark conditions in general. And as you can see, for uninfected individuals, they have this rest mode at night. But for the infected individuals, activity does not drop down that much. So we have this nocturnal restlessness. So even though activity in the dark is less than activity in the light, for infected individuals, it is still higher than their uninfected conspecifics. And then in the second figure, we go and explore the different time points throughout the day. So we have we have white for when the lights are on, and gray for when the lights are off. And what do we see here? So circadian rhythmicity and activity was present and distinctly different between control, which is the uninfected, and the infected individuals. So you can see here, they both have a circadian rhythm in their activities, this wave of motion, but for the infected individual, this has shifted, resulting in the infected individuals being more active at night. And as you can see here, throughout these different time points, the infected individuals have a higher activity. So this figure shows when the guppies are isolated or individual guppies, but then we also have a similar result for when guppies are in shoals, so in groups of 5. So you can see here the proportion of time spent being active in the dark is higher than the proportion of being active when uninfected. So the infected individuals, the infected shoals, are also restless at night compared to their uninfected shoals. And then I also mentioned we explored the rhythmical variance of the parasite itself. So parasite behavior and activity did not exhibit a daily rhythmical variance. But the interesting part here was that we also saw parasites being more active in the dark than light, coinciding very nicely with the infected fish behavior. So we then went on and discussed the implications of this study. A possible explanation for this restlessness at night could be the parasite infecting their host. So what happens when ectoparasites attach on the guppy host using their hooks? They also excrete digestive enzymes in order to break down the epithelial cells and feed on the skin of the fish. This causes some localized irritation to the host, and then they also start moving on the epidermis, causing further irritation because of the localized host immune responses. We then also went to talk about the complex interactions between infection and hormonal modulation. So we know that infection can cause the release of inflammation cytokines. And inflammation cytokines are directly associated with many hormones like melatonin, a hormone directly linked to rest activity cycles. So, perturbances in hormones like melatonin can further promote this restlessness. So, while infection can affect the hormone clock genes, perturbances in these hormones can also further affect the immunity of the host. These very interesting parasite behavior observed, where the parasite activity peaks at the same time as the fish activity, could be because of the parasites trying to infect new hosts, and this kind of dilating behavior where if an individual is very heavily infected, it will try and offload parasites to an uninfected conspecific, which will benefit both the host but also the parasite with this transmission potential. We also further explored the idea of chronotherapy, which is treating the different effects something can have at different time points throughout the day. So the use and application of chronotherapy to, let's say, maximize treatment efficacy could also be a potential solution. We've seen that there's it's not just light and dark, but also different time points throughout the day have a different effect. So how could that be used to our benefit? That concludes me explaining this poster, but you can scan the QR code to find the paper and more details about this study, but also you can find my email address and feel free to reach out and ask any questions you may have. Thank you very much.